to be here down in, well, for me, down in Sussex, because I'm living between here and London. But it's quite an easy place to get to. Also, my daughter's an undergraduate here, so nice to come down and have a chance to say hello to her. But really good to be here and be able to tell you a bit about some of our work with deep learning and NMR. So thanks for Itamar and Ivor for inviting me. So this came about really because I met Itamar at a conference in Padua in Italy, beautiful city there, and that was on NMR metabolomics. And he said, well, why don't you come down and tell us, you know, your AI folks here and something about this kind of work that we're trying to do at Imperial. So we are primarily an informatics methods group, but we are based in a faculty of medicine, mainly within a facility and a department which does what you might call omics profiling. So if you're not sure what that is, it's sort of, you come across genomics, I guess, you know, so measuring genes, transcriptomics, proteomics, and the really weird one that we do, which is called metabolomics, okay? Sometimes people find that difficult to pronounce, but thankfully after 25 years, I managed to do it. And it's all about, it's the science of measuring small molecules. So what we mean by small molecules is anything up to, say, 1,000 Daltons or 1,500 Daltons, something like that, in biological systems, trying to work out what the abundance of many of those different molecules, those different small molecules are in these biological systems, and understand how they affect and are affected by the biology that's going on. And we use different techniques to measure those metabolite abundances. One of them is NMR, and the other one is mass spectrometry. So anyway, a little introduction there, and I will give you a little bit more background on metabolomics in a moment. So I've got my laser pointer, which I hope. Oh no, now it's not working. Is that because I need to click on there, maybe? Let's try. Oh yeah, okay. Right, so before I start, I always like to give some thanks to the people who really did the work. So most of what I'm going to tell you today has been done by my PhD student, Jan, who has literally a couple of weeks ago had her viva and is in sort of a correction stage. And she also got married as well, so big life changes going through. And my collaborators, Beatrice, who used to be our NMR manager at Imperial, now working at the University of Adelaide, and Michael Judge, who's a postdoc with us, is now at NIST in the US. And also along with various other people who put a little bit in as well from time to time. So, oops, wrong way. So here's the plan of what I want to talk about. I'll give you a little introduction to what metabolomics is in a bit more detail, and also some of the sort of interactions with the deep learning side. I know that some of you are more AI experts, some maybe slightly more on the chemistry side. I don't know. Actually, maybe we should have a show of hands. Who here would consider themselves more on the computing and AI side? Okay, and who would consider themselves more on the biological or chemistry side? Okay, all right. So we've got a slight majority there of more the AI side. So I hope I'm going to be able to explain the sort of chemistry and the metabolomic side well. But if there's anything that you don't understand, please just ask, okay? So we'll have a bit of introduction there. And then there's a couple of stories I want to tell you. One is about enhancing resolution of a particular kind of spectrum that we use for profiling these metabolites in biological mixtures, which is called J-res NMR spectroscopy. And then the second story I'd like to tell you, which is only a partial story because it's not published yet, is about, again, using another kind of deep learning approach inspired by video peak tracking to look at peak tracking in the same kind of spectra. Gosh, I'm really not used to this pointer. So the first part, the introduction. So what is metabolomics? This is my metabolomics in one slide introduction. It's the study of the complement of small molecules within biological systems, which I already told you. Please do come in. So if we have a biological system represented very abstract here, so we've got an organism. Within an organism, there are biofluids. And within those, we could have tissues, and within those, we can have cells. So we know that biological systems are built from cells. Inside the cells, we have lots of different kinds of molecules. We have DNA, we have proteins, And we also have these small molecules, which we call metabolites. So again, up to say 1 or 1,500 mass units, if you like. And those are the primary types of molecules which sort of make biology work. In particular, metabolites are the building blocks of these larger molecules. Both DNA and proteins are built from the building blocks which these metabolites provide. Either they are the building blocks themselves, or they're involved in reactions that produce those building blocks. Metabolites also have other functions, such as providing energy to the cell. They're like an energy currency. So think of ATP, you may have heard of that. It's an energy currency to provide energy to reactions in the cell. And they also have other roles in biology, like signaling. But we don't have to worry about it too much, the specifics. The main thing is that they are involved in a huge number of biological processes, if not all biological processes. And so they are metabolites as a whole. There are many thousands of different metabolites in a given biological system. They're influenced by, and they influence many other factors. So lots of things that we're interested in biomedical science, such as the effect of diet, drugs, genetics, age, disease, of course, they will influence the levels of these metabolites inside cells and inside the other compartments of the organism, like the tissues or the biofluids. So if we take samples of those biofluids or the tissues or the cells themselves, we measure the levels of those metabolites, we can get an idea a bit more about what is going on with these different factors which are biologically and biomedically interesting. So how do we measure those things? I haven't got used to this still. We take what we call metabolic profiles. Okay, so for us, a metabolic profile is really just a list of metabolites and their levels in a given sample. So, certain concentrations for different molecules. And the data itself that generates that, as I mentioned before, is either generated by NMR, so nuclear magnetic resonance spectroscopy. Here's an example of a typical NMR spectrum. And the other technology that's widely used is mass spectrometry, often preceded by some chromatography. So liquid chromatography, mass spectrometry is another technique, which I'm not going to go into at all here. But the primary thing about metabolomics, and it's the same with other omics in the omics fields in general, is we try and measure as many things as possible, and we don't at the beginning have a hypothesis about which of those molecules will be involved in the disease. So if you can imagine genomics, for example, we might want to look at the entire sequence of an organism and work out which parts of that sequence could be related to a particular biological process or when a biological process goes wrong. We don't a priori select a particular gene and say, it's definitely that one. And the same thing happens in metabolomics. We measure as many metabolites as possible using these techniques, and we let the story emerge from the data by using usually some kind of advanced statistics or machine learning, or in this case, some deep learning. Okay, so what can metabolomics do? Well, I just did a quick sort of Google search basically to show you a few applications. So one of the biggest areas, especially in the biomedical area, is it generates biomarkers and an understanding of how diseases operate. So here are a few high-impact papers. So, for example, cancer biomarkers for cancer, which have actually been developed into clinical biomarkers, steroidal biomarkers for more different kind of tumors there. And there's a big focus of research on cardiovascular diseases, because cardiovascular system requires a huge amount of energy to be generated, and the metabolites are involved in that. So producing biomarkers of disease potentially for clinical tests is one application of metabolomics, but there are plenty more. Outside of biomedical science, there's things like plant sciences, so it's telling us how tomatoes ripen. Might sound a bit esoteric, but actually a huge industrial involvement. A lot of agricultural revenue depends on fruit ripening at the right time in the right place. Marine biology, so this is all about coral reefs. So I guess the people who did that had a very nice trip to somewhere to swim. Food science, big area again. This is about anaerobic fermentation. So how do metabolites get involved in those processes that may produce something that's of relevance for either the food taste or the food texture, or in other areas, biotechnology, which I've got here. So how can we actually produce certain small molecules on an industrial scale by, for example, re-engineering bacteria to produce a certain molecule that's of interest? So what do metabolic profiles look like? I showed you some very tiny earlier on, but this is essentially our primary data that we start with in metabolomics, at least on the NMR side. So this is a one-dimensional, we call it one-dimensional because there's one frequency axis here. So this is frequency. It's a spectrum of human urine. In metabolomics, we deal with a lot of horrible sort of human biofluids. So apologies for talking about that, but that's what we deal with. And you can see there's many, many different peaks here. Each of those peaks will correspond to a signal from one metabolite. Okay. Now, each metabolite can give rise to more than one peak, but the main message I'm trying to convey here is that there are many, many different metabolites present, perhaps hundreds to potentially low thousands worth of signals, giving us information on tens to hundreds of metabolites. And if we look at the other kind of data, the mass spectrometry data, you can see again, it's a very complex kind of data with many different peaks, again, telling us which metabolites are there. Mass spectrometry is a bit more sensitive, so we can get information on more metabolites. But we use both of these techniques. And the big question is, keep forgetting which way is the right way to go forward. How do we extract information from that data? Okay, you can see it's very complicated. There's a lot of overlap between these peaks, and we need algorithms and statistical methods to be able to extract information in a way that is useful to research scientists. Now, I thought deep learning for a long time could actually have a good impact on this because, it's in some way doing some magic. And so we thought that maybe we could go in this direction. So I had a look to see, what people have been doing in metabolomics with deep learning. And this is a kind of embedding of lots of different publications from the metabolomics field, colored by the different types of sort of statistical and machine learning approaches that are being used. The pink ones here are multivariate statistics. Blue is machine learning, and brown is where you've got deep learning on neural net. And you can see that actually it's a rather small number that people have actually been applying deep learning in the metabolomics area. If you look over time, this is multivariate statistics as a percentage of total papers in metabolomics as a keyword, gradually going down, say, over the last 10, 15 years, whereas machine learning and deep learning just beginning to improve there. And I think we'll see things going up a lot. So the message there, I think, is that metabolomics is an area with lots of data, lots of complex problems where deep learning and AI could make a big impact. Things that people have actually done with deep learning in NMR-based metabolomics, I've put on the slide here. Now, I don't want to go into details here because it would take too long, but there are various different types of deep learning network that have been used. So all the usual things I'm sure you guys are familiar with. To solve some of the basic problems. So things like, how do we find peaks in those complex data? How do we match a library of known compounds to our complex data to see which compounds, which metabolites are actually present there? That's a really fundamental problem. We also have done something which I'm going to tell you about, which is around improving resolution. But ultimately, most of these applications of deep learning are around finding what is in the sample and trying to name it, give a chemical name to a metabolite. Here's one example, which is the peak picking one that I mentioned. So here's a very complex two-dimensional data from an NMR spectrometer of, in this case, mouse urine. So again, you can see lots of different signals, this time represented by contours. And what the deep convolutional network here has done is try to find where it thinks there are reliable peaks in that data, which at first sounds like a really trivial problem, but actually because the peaks overlap so much, it actually becomes quite difficult. If we look in one-dimensional spectra, this is the kind of output it can give, and just demonstrating that it can kind of deconvolve very highly overlapped patterns. So that was a bit of an introduction. Now I'm going to go to talk about the first story, which is our efforts with these JRES spectra, which I'll explain in a moment, to try and improve resolution. So that's getting, if you like, more information out of the raw data than we would normally be able to get just by processing in the standard way. And this has been published in the journal Analytical Chemistry, and so there's a QR code there, which I'll show again at the end if you're interested in going back to that. So for this work, we've been using generative adversarial networks, so GANs, again, probably very familiar to those of you in the AI area, but just to explain it to anybody who's not familiar, a GAN is essentially is quite a really fun kind of a network setup where you have two networks, actually. The first one is the so-called generator. And what the generator does is it produces new data for you, okay? So an example could be we're trying to produce images of faces, for example. So we want to build a network that can produce images of reasonable plausibility of human faces. And in order to do that, it needs some kind of input, which can be just noise, okay? So we can give it some random numbers, essentially, and it can produce something which we hope will look like a reasonable face. But obviously, setting up a network to do that right at the beginning, random weights, etc., it's not going to produce you a face. So how do you train it to do that? So what you do is you compare it, or you compete it against another network, which is simultaneously trained, which tries to tell the difference between the artificial data that's being generated by the generator and real data. So you give it some real images, in this case of human faces, and the discriminator has to tell whether these ones that are presented to it at random, if you like, are real or fake, okay? And what you do is you train these simultaneously with various loss functions so that the generator is getting better and better at generating these plausible images, and the discriminator is simultaneously getting better and better at discriminating between the real and the fake data. So in the end, you hope that you have a generator that is super good at generating plausible images of whatever it is you're trying to do, and with some kind of input, you can then produce new data, in this case, images of faces, which are plausible. Okay, so how does that apply to NMR metabolomics? Okay, you might be wondering why faces makes any difference. So let me just provide a little bit of background on this particular type of NMR data that we use. So this is a so-called J-resolved spectroscopy. It's something that gives us a high degree of information about the structure of the molecules, the chemical structure, so which atoms are bonded to which atoms. And sort of technicality, I suppose, if you're not in NMR, that what it does is it separates chemical shift information from scalar coupling. But it's probably best just to look at something like this. So this is a spectrum of a small molecule. The structure is down here. It's 4-hydroxypropiophenone, okay, just an example. And you can see that there are two axes. Both of them are frequency axes, essentially. And what we're showing is the intensity of the peaks as contours. And you can see that there's a very specific pattern, okay? We've shown this sort of profile, projecting it onto one axis along here. And you can see that there are various peaks at various positions. And essentially, the important thing to take home from this is that the pattern of the peaks, the positions and the intensities of the peaks are characteristic of the structure of the molecule. So if we have a different molecule, different small molecule with different structure, we will get peaks in different positions. The other thing to draw your attention to is that it is symmetric about the 0 Hertz line on the y-axis there. So there's a y symmetry there, which is something that we then take on later to build into our deep learning network. So that's one molecule. When we look at a complex mixture such as human blood or urine or something like that, we get a mess, okay? So this is now, if you like, the sum of lots of these different molecules all added together at different weights, if you like. So we've got lots of different peaks all over the place, many of which overlap with each other, okay? And that's the key point, that it's a very, very crowded spectrum. What we'd really like to do is improve the resolution of that. Now, there are experimental ways of doing that. We can go to higher fields of the NMR spectrometer. We can do some other clever things with the NMR. But in general, there's only so much you can do, and it's expensive. Okay. So, oops, wrong button. That's what we want to do. We want to deconvolve or try and separate as many of these crowded signals as we can. And that's the same thing as saying we'd like to increase resolution. Okay. So this is the typical sort of workflow you might have in a metabolomics experiment. You prepare some samples, you put them in the NMR spectrometer and you acquire some data, then you have some processing, and you end up with beautiful process spectra you can then go forward and analyze statistically. And there are ways in which you can improve the spectral resolution at any of these stages that are already existing, okay? So there are things called non-uniform sampling, which we can do. We have to adjust how we actually acquire the data in the lab to do that. We can take already acquired data and do something called linear prediction, which is a very straightforward, like a linear regression kind of approach, to try and estimate what the spectral shapes would have looked like at higher resolution. And then there are some other methods that have been around more recently, essentially trying to fit statistical models to try and estimate what the spectral shapes would have looked like at high resolution. But they all have disadvantages. So these things require you to do something special in terms of the lab work, for example, or they have kind of varying performance, not very high performance in terms of really improving resolution. And some of these other ones are quite high intensity in the sort of computational. You need a lot of prior knowledge to be able to run them. So all of them have drawbacks. And so we, of course, thought that maybe some of the deep learning approaches that were used in image analysis out there in the rest of the world could perhaps be used to improve resolution of these highly overlapped and crowded NMR spectra for metabolomics applications. So this is how we applied it. So we're not deep learning specialists, so we took something that was relatively off the shelf. There's a network already out there called Real ESR GAN. Okay, so it's a generative adversarial network for enhanced spatial resolution. It has these two parts, the generator and discriminator, which work together, as I've already explained. And if we have images, we can input, once it's trained, we can input a low resolution image and we can output, hopefully, a higher resolution image where everything's more clear. So what we want to do in our NMR metabolomics application is replace those images by NMR spectra. So this is a small part of that two-dimensional map that I showed you before from the NMR spectrum. This is what we're calling our low resolution spectrum, LR, and it will produce a so-called super resolution spectrum, so the predicted higher resolution spectrum from this trained generator. So that's our plan. Because we base this on ESR GAN, we called our method JRESA-GAN, okay, just to sort of put those acronyms together, okay? And the key thing was we weren't just taking this as off the shelf completely. We changed the loss function, particularly to take account of this Y symmetry, so the symmetry between the top and the bottom half of the image, to try and exploit that from the point of view of metabolomics. Obviously, with deep learning, you need a lot of data to train these very complex networks. That's a big problem for us in metabolomics, because although we have quite a lot of data, it's usually not very well labeled or not reliably labeled. So what we did in this case was we simulated data, okay? The student had to build a simulator for that, but essentially we ended up with able to generate thousands of pairs of low resolution and high resolution complex mixture spectra. So similar to the kinds of spectra that we would get from human biofluids, for example. And we actually tried to simulate a specific biofluid, human urine, to do that. So the way we did that was we took individual spectra of known compounds and just added them together electronically, if you like, to produce a mixture spectrum. And that produced us a lot of training data at high resolution, so the highest resolution we could have. And then we artificially blurred this data using a convolution function to get a lower resolution representation of the same thing. And so what we're providing to the network to retrain it is these pairs of HR and ALR, high resolution and low resolution mixture spectra. So that's the training data. We use this thing we call Metasimilo, which is an app for simulating realistic NMR spectra. If you're interested in that, from maybe only the NMR people that would be interested, but I can talk about that later. So we've got the training data, and of course we need some test data as well. For this, we decided that we would make it a little bit more challenging, and instead of simulating the same biofluid, we instead simulated blood plasma as a test data. So we're training it on human urine simulations, testing it on human blood simulations, so to make it a bit more challenging. But essentially the same idea applies. We add together spectra from pure compounds where we know what they are, but using the ones which are present in blood rather than urine. And we also tested it, of course, on real experimental data. In this case, we've got various different kinds. And again, for the non-NMR people, don't worry about this. It just means that we have got pairs of low and high resolution experimental data. In this case, there's one where we're so-called 40 and 80 increments, which are different resolutions of the JRES And we've also got a different pair where 80 becomes the low resolution and 160 becomes the high resolution. Okay, but don't worry too much about that. We also needed some metric to decide whether we were improving the resolution or not. To measure that in a quantitative way. We just came up with a very simple metric, which depends on measuring the height of two peaks, the two nearest neighbor peaks in the spectrum, and looking at the depth of the valley in between. Okay, so we came up with a resolvability score. It's nothing groundbreaking, where we essentially look at the ratio of the depth of this valley to the height of the two peaks. Okay, so higher resolvability score means better resolve. And so the results I'm going to present you are going to be presented in this so-called, what we call a V-plot kind of form, okay? So we're plotting resolvability on the y-axis, and then we have the original data, high resolution, the original data at low resolution, and then the predicted super resolution data from the network, okay? And this is the ideal profile for what we would like to see happen. We would like to have high resolution data, low resolution data, and then the predicted high resolution or super resolution data should be, of similar resolution or better than the original high resolution data. Okay, so we like to see these V kind of shapes. So this is on simulated data, again, trained on urine and tested on human plasma, and you can see that, well, it does pretty well. So each one of these lines connects one pair of peaks in the data, okay? So each peak might be in there more than once, but each line is a separate pair. And you can see that in almost all the cases, we have this V shape where the high resolution data is well resolved. We go down to less well resolved peak in the low resolution, and then the super resolution prediction is recovering essentially what we had in the high resolution data. So that's all great. We can plot it in a different way if we compare the resolvability scores from the high resolution data, this one, to the super resolution data, the green one here. And you can see that pretty much, it's recovering not too badly the resolution that we had in the original data. So that all works nicely, but of course that's simulated data, and of course it would work, wouldn't it? I can show you some examples. So again, in the two-dimensional sort of image domain, you can see a high resolution peak cluster goes down to low resolution. You can see that you've completely lost the fact that there are two pairs of related peaks here, and then you're recovering it again in the super resolution prediction. So that's all good. Here's one that doesn't work quite as well, but still not too bad. You see the original and the predicted look really similar. And then, of course, there are always some situations where you don't pick everything up. So, for example, here there are actually two peaks in the middle, and we still don't find those two peaks. So, there are some problems that even the deep learning approach can't recover. But the real test, of course, is experimental data, because that's what we want to use it for. Again, we're testing it on blood plasma, so trained on urine, but testing on blood plasma. And this is the kind of V-plot we get. Now, you can see it doesn't look quite as nice, all right? But still, for a large proportion, in fact, 65 out of 68 peak pairs are showing a higher resolution, a higher resolvability in the super resolution than the low resolution data. And you can see some various different kinds of changes in resolvability for peak pairs between high resolution and low resolution. That's often due, either they are going in the right direction, getting more poorly resolved in the low resolution, or if they're not, if they're flat, or if they're going in slightly the opposite direction, it's due to peaks being very tiny and the effects of noise. Don't forget, the low resolution and high resolution here are actually two separate experimental acquisitions. So the effects of noise and a few artifacts and things are different between these two. And you can see that we get the similar sort of plot comparing the high resolution and the predicted super resolution here, but actually quite a lot of the peaks are even better resolved than you might expect. Okay, so at the end of the day, this is something we thought actually is working not too badly, even on real data. The key point is that to go from this low resolution real data to the high resolution data might be going from something like a 10 or 15 minute acquisition up to something like 3 hours on the real spectrometer. So it saves, if you're going to rely on the predicted super resolution data, you're saving a huge amount of time. In metabolomics, I didn't mention, but we're typically running hundreds or maybe even thousands of biological samples on these spectrometers. So if we can cut the time by a factor of 5 or 10, that will really make a huge difference in what we can practically do with a big study of many people and clinical applications, for example. Here are some of the real data, again, sort of zoomed in on particular examples. Here's a nice one where you can see quite a complex peak structure, lost in the low resolution, but again, reasonably well recovered. Here's one that doesn't look quite as good, but not too bad. We're even recovering those very closely separated peaks in the middle there. It doesn't just work with human blood plasma. We can work with other, oh, that's the next slide, sorry, but I was just the NMR people to show that it'll work at different field strengths. So this is other data sets from the same spectrometer frequency. So this is the same magnetic field strength that we had in the original data. It's a bit difficult to see here, but essentially 80 to 90 or more than 90% of them show an improvement in resolvability. But if we take exactly the same network without training and apply it to a different, completely different field strength, in this case, a higher field spectrometer, again, we're seeing quite good outcomes there. And what I was just talking about was we can apply it to different kinds of data. So not just human blood, for example, but this is milk. Okay, so there are food applications of metabolomics where milk is a very complex fluid itself. Again, you can see that there's very strong performance in terms of resolvability there. And this is orange juice, okay? So plenty of applications for the same kind of techniques, not just in human or otherwise biofluids, but in food and other areas. There are some drawbacks, of course, and one of the questions we obviously had to ask was, well, we're predicting these high resolution data, but how reliable actually are those peaks? You know, is it coming up with hallucinations, or is it losing peaks where it should have peaks? So what we did was we compared really high resolution data that we took as the ground truth with the prediction from taking the low resolution data and using the network. So in order to detect peaks, you have to have some kind of threshold where you cut the data to say, yes, anything above a threshold is a peak. So this is what we've got on the x-axis here. So different thresholds, higher threshold, smaller threshold. And what we're comparing here is the number of true positive peaks. So peaks that actually match between this high resolution, almost perfect data, if you like, and the predicted super resolution. And you can see for a high threshold, we do get an increase in the number of peaks. The total number of peaks detected is low because that's a very high threshold. As we go down to low and low thresholds, we're still getting an improvement in the number of true positive peaks. So we think actually, you know, most of the peaks that are there are fairly reliable. But if you look at all the peaks that are detected at these three different thresholds, so now not just the true positive, but all peaks, the total number of peaks detected, you can see that, yes, the number of peaks detected is higher at all these thresholds. But what I plotted in purple here is the number of peaks that really match up with the true positives, with the high resolution experimental data. And you can see that while at the higher peak threshold, so larger peaks, you're getting quite a reasonable number of those peaks being reliable. At the lower end, where you've got quite noisy peaks, you are finding some suppression. So the network is actually improving the resolution, but it probably is suppressing some of the smaller peaks, even genuine peaks in the data. So it's not perfect. Okay, so to summarize this bit of the story, JRES, it seems to be the JRES, we've implemented these specific improvements using this kind of symmetric loss function into this off-the-shelf resolution enhancer. It seems to do reasonably well. It's quite promising. You know, it hasn't been used everywhere yet, but we showed that it can work at different field strengths and sample types. There's no special experimental analytical workflow required. The users can just take their normal workflow and apply it to the data that they already acquired. So they could even go back 20, 30 years, take really old data and improve the resolution of that, which is a really huge advantage to compare to some other techniques. And it can also be applied in combination with other resolution enhancement techniques if you want to. But of course, there are some limitations. As I've mentioned, low-level peaks potentially might be a little bit suppressed, and we haven't really evaluated how quantitative it is. So exactly, you know, is there a linear response as a compound changes in concentration? Is there a linear response in the height of, or the area under the curve, if you like, of the resolution enhanced data? And of course, you could apply the same idea to lots of other kinds of NMR or in fact other instrumental data, but you probably will need to retrain. So that's the JRESIGAN story. And for the last part of the talk, I just wanted to give you a shorter, an incomplete story, looking at a different type of deep learning applied to peak tracking, but also from JRES spectra. So the same kind of data going in, but trying to solve a different problem. So this one is the problem. Sorry, yeah.
Quick question.
Yeah, go. Yeah.
Just on the maybe I missed it, but what was the symmetric loss function?
Yeah, okay. I realized that I probably should have explicitly put that in. So we experimented with a few different symmetric loss functions, but we ended up with just something based on a correlation approach. So a correlation between the top and the bottom half of the data within each column of the spectrum. Okay, so something really, really simple. And we had to do a bit of rejecting of zero values and things like that. So we don't use the entire spectrum, but we use where there are existent data. Because in these JRES spectra, even though they're quite crowded in the middle, there are areas at the edges that are quite empty. Yeah. So, and we just add that on with a weight to the original losses for the GAN, which are like the discriminator and generator losses.
And are the data, are they exactly symmetric, or is there differences in like noise and things like that?
There will be differences in noise. We haven't explicitly taken that into account. We don't estimate the noise, for example, but because we're tuning the weight parameter, that does account for that to some extent, right? But yes, there are both noise differences, but also in real data, you tend to get some artifacts. There are various processing things that you have to do which can introduce artifacts themselves. So we had to do a few small tricks, but nothing crazy. I mean, like we ignore the very edge bands of the spectrum, for example, where you do get more artifacts. So yeah, there are some changes, but essentially we're just trying to make sure that the generator will produce symmetric predictions. Any other questions at this stage? Yeah.
Why don't you just open them?
Yeah, can do. So yeah, we actually didn't try that, but we decided that because this was a particular characteristic of the data we were going to include in the loss function, we felt that that would be more interesting to see if we could incorporate that into the training than to just treat it as a standard image without any of this extra characteristics. So always like to try and exploit any particular characteristics of the data where we can find them, rather than processing the data which removes that characteristic, which then becomes more of a normal problem. We wouldn't be able to produce a new network which would do anything with that. But yes, it's probably something that we should have compared it to actually, but we haven't done that. Good idea.
Why wouldn't that just be done normally, just when you're doing your...
One reason is because of convention. Essentially, the way NMR spectroscopists look at this data, they like to be able to see the symmetry. Okay. Another aspect is to see that the data are so it's like quality control of the data, I suppose. So if there's any areas which are not symmetric, you know that they are probably artifacts, etc. In theory, you could fold those over once you've done all that quality control, and you could probably look at that. But it's the way that NMR spectroscopists always view their data is with this symmetric in there. Perhaps Itamar or one of the other NMR people can add to that.
An NMR person.
Oh, okay.
So would it give you an FNR increase, like a group 2 increase, or having that data spread out to that sort of count for that anyway?
Well, the same amount of information is there, right? I don't know for sure off the top of my head. I mean, I think, you are fitting double the amount of data in some way. So I would imagine that in terms of signal to noise benefit, you're not going to gain anything, because one way you're just adding together those random errors. In another way, you're fitting double the number of data points. So my guess is that you probably wouldn't gain anything. I mean, one thing that might gain you more is to use, so I didn't mention, but in NMR, well, you probably know that we have real and imaginary parts of the spectra. So you could think normally people completely ignore the imaginary part of the complex spectra, but if we fit that as well, then that could be something that we could potentially improve signal to noise. But one thing I should emphasize here is we're not trying to, or we haven't explicitly tried to improve signal to noise. Okay, what we're trying to produce is an improved resolution. So even if the signal to noise is a bit lower, we're okay as long as we've managed to separate peaks that would otherwise not be separated. At least in metabolomics, high field NMR, the signal to noise is not usually a big deal. Occasionally, we want to look at
It
very
would be
small
when we
things,
go
but it
to
can be.
the future benchtop.
People have tried in metabolomics a little bit to try and improve signal to noise via some of these approaches. It's become a little bit controversial because you end up with essentially kind of fitting a known peak function implicitly or explicitly, and then you almost say that, well, there's no noise left because I fit this thing. So, you know, what are you really doing to the signal to noise is a bit more tricky. Okay. Yeah, there's a question.
Did you use a GAN just because there was a model already available for that? Or why did you specifically use the GAN?
That's a large part of the reason. So that ESR GAN came out, I think, in 2021, and my student was starting just after that, and we thought it would be a really interesting approach because it seemed to be doing, you know, it seemed to be really at the forefront of these image processing and resolution enhancement techniques in natural images. And certainly now there are other architectures that maybe outperform that, but this is something we started when this was one of the leading techniques. But we didn't do a huge search of, you know, comparing different architectures. Okay, yeah, yeah, well, yeah.
And how exactly do you know the kind of blurring function that you applied? Because you had some downsampling and blurring.
Yeah, so what we're trying to do is replicate the difference in resolution between real data taken at higher node resolution. And I mentioned this number of increments you have in the pulse sequence to get higher or lower resolution J-res spectra. So we could actually, you know, in theory, you can predict that it actually should have a certain difference in the line width, if you like, or you can measure it in the actual experimental data taken in two different ways.
Tricky because the number of increments is just truncating the data. And truncating the data, you can do two things in order to get your full resolution spectrum. Is either if you want to avoid the Gibbs fluctuations, you just have to weight down by some sort of exponential decay, and then you artificially broaden the data. But in itself, the truncated data doesn't have a less resolution physically, just less digital resolution. Right?
No, I mean, it's more the practical result of doing that. But at the end of the day, you know, in terms of deciding what kind of blurring to apply, we were simply comparing the real data, you know, how much wider are the peaks in one than the other. And so it's easy for us to estimate what kind of blurring to put in. We did experiment with different blurring kernels and things, and it actually didn't really make much difference whether it's a Gaussian kernel or a PAT or something like that.
I'll come back to you with
lots
of questions later. I'm really curious about the last story.
Okay. All right. So as I say, let me go on to the second story. And as I say, this is not public. The previous one is published, but this one's not published, so it's a bit less detailed. But I thought it's a nice another sort of idea of how some deep learning approaches can maybe help in this area. So this is the problem. Here are three actually simulated NMR spectra of blood plasma, I think it is. And you can see that there are many peaks, okay? You've got this symmetry on the y-axis, as you can see. And if we take particular peaks from particular known compounds here, and we look at these spectra under different physical conditions, so these are different pHs that we've simulated, so different acidities of the sample, and you can see that the peaks that are here are moving around, okay? So they're not present in exactly the same position in each simulation. And that is something that happens in real data all the time. Even though, as a chemist, you try and keep the pH as constant as possible, even small variations, smaller than we see here, will produce small variations in the peak positions, even for the same compounds. And that seems like a minor thing, but actually it becomes a huge problem when we're trying to statistically analyze these data. So, you know, what we might be doing is comparing one sample to another and seeing what's changed. If the change is actually just a change in position because of something like a change in acidity, that's actually not very interesting change for us. What we're looking for is which peaks, which metabolites have gone up or down in concentration. So we would really like to be able to take out this positional variation as much as possible, or at the very least, match the peaks between these different samples so that we can say, yes, these two peaks you can see are the same ones in each one, and maybe these ones are the same as those ones there. So there's a peak tracking problem that we have to address. These are simulated data, which are obviously relatively simple. When you look at real data, it becomes even more tricky. Now, you can do this manually. There are alignment algorithms that can be semi-automatic, but usually have to be tweaked manually. But it's pretty time-consuming, and it's certainly not perfect. So we, again, wondered whether maybe we can apply some deep learning methods to this. And we came across this technique called YOLO. Anybody come across that? Yeah? Okay, I'm sure it's pretty widely known in AI area, which is an object detection and tracking network, basically, or a series of networks. So it's been around for probably 10 years or something like that under various versions. And what this does is it tries to take a video sequence, so a sequence of images. It has two parts to it. So the first part is a detector. So it's trying to find objects within these natural scenes. So here, if you take vehicles on a motorway, it's trying to find where the vehicles are. Okay, so put bounding boxes around them in each of the frames. Okay, so that's the first step. And then the second step is to try and track them. So to work out which objects are the same ones between these different frames of the video. So the tracker does that. And you end up with a set of trajectories, not really well shown here, but basically we can say, yes, this truck is the same one in two different images, or this car is the same one in two different images. And we thought, well, that's pretty much exactly what we're trying to do with our J-ray spectra. So if we take the analogy between these two situations, so instead of frames in a video, we have samples in a biological study. So say 20 individuals, each one gives a blood sample, each one of those becomes one image, if you like, of a J-res NMR spectrum. Okay, so we've got a sample sequence. We have objects, which in this case are vehicles or something in a natural scene. And of course, we have what we might call multiplets, so just the peaks linked together by their pattern in the y-axis. And we're trying to detect those. And then we have different types of objects. In this example, they're different types of vehicles. And in the J-res NMR situation, we can classify our peaks by different types. They're either a single peak in the center of the spectrum, so for example, like that one, or they have a different structure on the Y domain with 2 peaks or 3 peaks or 4 peaks or more peaks, etc. And those are very characteristic of the type of chemical structure. And as I said, it's a pattern that we would like to maintain and recognize to be able to understand which molecules are present, okay? So there's a lot of analogies between these situations. So we thought we would try giving this YOLO architecture a go at treating our NMR data. So, but obviously we didn't want to just take it completely off the shelf again. We wanted to implement some changes that made it more specific. Again, we trained it with simulated data, so using our built simulator. And we did some data augmentation this time. So again, for the IEI people, you know what that is, just increasing the training set by applying some transformations to generate new training data that is also realistic, different examples of the training domain. So there are various standard augmentations like taking peaks and moving them, translating them along the x-axis or something like that, which is fine in NMR to some extent. We also introduced a J-specific augmentation. So this, when I say J, I mean the y-axis of the data. Again, you can see that we have this symmetry in that area. Different chemical structures will lead to different splittings, different distances on the y-axis between these peaks. So, for example, we might take a real image and generate an augmented spectrum or image by moving those peaks apart by different amounts. So you can see that essentially these 2 peaks have been moved a bit closer together. These 4 peaks have been moved a little bit closer together, at least in the middle. And that would represent a different kind of chemical structure, okay? Now, what we didn't do here, so this is the transformation we made, what we did not do here was try to apply any actual chemistry rules to it. We don't know that these splittings, these distances between the peaks actually represent a real molecule that could exist. We could have done that, but it would have been a huge amount of extra work to check that all works. So in this case, we were just applying essentially random movements that were still symmetric in the y-axis. So that's one of the things we did. We also implemented the symmetric loss, which we already talked about for the J-Resogan application, in addition to the the other loss functions that it already has. So we have a retrained detector, and trying to look again at simulated data to see if this works, so to see if it can be detected. Here you have the ground truth. So this is peaks that have been manually annotated by spectroscopists in this very complicated, still simulated data, but it's still quite complicated. And here you have the actual detections from the YOLO network or modified YOLO network. And you see that actually almost pretty much all of the peaks that are present in the ground truth are detected, okay? Now, there are some overlapping areas, so very crowded regions here where we're still detecting separate peaks, but it's not perfect. So sometimes we miss a peak because the detector thinks that it's actually, two peaks are actually part of one because they're quite overlapped. And also, oops, I thought I had it on that slide, but also we may not get the exact peak class correct. So where we think it should be a 3-peak multiplet, it may say it's a 5-peak multiplet or something like that. For our applications, although that is interesting, it's not the primary concern because our primary concern is to track the peaks. So that's the first part, that's detection of the peaks. And the second part is this tracker, okay? So the tracker traditionally has a Kalman filter. Again, for computer scientists, I'm sure you know what that is, but essentially an algorithm predicts the position of an object in the next frame of the video from the position in the current frame. And for us, that means taking one sample spectrum and predicting the position of a multiplet in the next sample spectrum. And obviously, it needs to be evaluated by wondering if the position is correct. And that's the standard way that this so-called bot sort tracker that's incorporated within YOLO works. But we also added something which would look at the shape of the peaks. So in NMR, it's really important to have the peak shape, particularly the multiplet structure, as I mentioned, how many peaks are there in that multiplet and what is their relationship to each other in terms of the peak ratios and things. So we added a correlation, again, a metric that looked at the profile of the peak along that y-axis to see if the correct peak was being tracked, essentially. So we say both is the position of the peak correct, according to the Kalman filter prediction, and we also ask whether the shape is correct. And that's how we decide whether the multiplets are being correctly matched. And again, here is just, in this case, it's a video of about 20 simulated NMR samples. And if we just let it run through, I hope it's going to do something. Yes. So it's going through a set of samples one by one. And you can see that if we just take this peak on the left here, you can see that it's quite hard to see actually on this projector, but it is being detected. You can see that the color of the detection is changing, which means that it's not always getting exactly the same multiplet type correctly predicted. For the most part, it's okay, but occasionally it gets it wrong. And then if you take something which is a bit sort of crowded, you can see there's a 3-peak multiplet here, which is moving around. It starts off, if we just wait for it to go back to the beginning, starts off over here, and then it moves gradually actually through another peak, which has 2 components. And you can see that it is being detected okay. It's being tracked across that thing, but when they completely overlap, it's being lost. Or one of them is being lost, and sometimes the tracking IDs are confused as well. So it's working to some extent, still not perfectly. We'd say that probably most of the multiplets can be tracked correctly. In some cases, it does get misclassified, although, as I say, it's the tracking that's most important, not the classification. And we certainly have areas where when peaks overlap in a sequence, they can be lost. But that's something we would like to add on later as a kind of interpolation prediction. So that's all I have to show you in terms of results of that. It's to be continued. So we are still looking at validating this with real data and enhancing it, but it's something that we think could be quite useful in the future. So I'm going to finish there and just give you a quick summary. If we think about deep learning and metabolomics, I mean, I would say it's kind of ripe for these things. What we're lacking is more labeled, thank you, more labeled training data, particularly labeled data, but for some problems that can be addressed by simulation. The generator GAN story, we can do something quite nice with improvements to the ESR GAN network that's already out there. It does give you promising resolution enhancement, and it can work with lots of different types of real data. And finally, for the peak tracking in the J-res spectra, this is something that's only just starting and it looks promising. And we're hoping that what we can do is track the compounds well, so that then later on we can annotate them, which means identify the compounds and potentially quantify them, producing a kind of automated pipeline for practitioners to use in NMR metabolomics. So with that, I'll just leave you again with the QR code I mentioned I would put up later, and say thank you for your attention.
Yeah, thanks for the talk. It's really amazing. I can understand some things which I'll have to learn.
Great.
Yeah, I've got two questions. One that's smart and one that's dumb. The smart one is on your tracking experiments. Have you considered using optimal transport to do that instead of ELO?
Do you mean as a method or as a metric to, yeah? Well, obviously it's something I've come across in the literature, but I haven't experimented with it yet. I honestly don't know much about it at all. So thought about, but not very deeply so far.
One of the nice things about is that you can quite naturally encode structure into that transformation. So you can preserve, if you're making a map between two, you know, distributions, you can preserve the topology, the maps in that transformation. So it could be quite suited to this. And I know that it's been applied in other omics analysis quite recently. The dumb question is to your original, and there is actually, sorry, a connection to GANs as well. In the metric, but in your original analysis, this resolution enhancement, and this is obviously something that you, as a non-expert, must be considered before. But if you've got a reasonably good idea of the width of the peaks, can't you just threshold? That is a really naive approach. Can't you just put some threshold, find peaks, and then to try and split peaks where you've got two very close to each other, you say, oh, well, another threshold, and how broad the peak can be. If it's above that threshold, maybe there's two peaks there. How well does really simple like that work?
So it works to some extent, but not quite a long way from perfect, I suppose, is sort of the basic answer. I mean, it's essentially a deconvolution problem. And people have worked on this. I mean, I've worked on it as a postdoc using Bayesian approaches to deconvolution, and other people have done lots of things. And I think the problem is that although we have, and a spectroscopist will tell you that, you know, their peaks are all well characterized and they get the right peak shape and the peak width all the time, in reality, that's not as controllable as people really want it to be. And so even within one sample spectrum, you'll have small variations in peak shapes and peak widths. And certainly from sample to sample, running, you know, if you run 1,000 samples across several weeks of time on an NMR spectrometer, even though it is actually a really stable instrument, you can still get small variations. So, you know, there are limitations in what you can assume in terms of peak shape and stuff. I mean, and another aspect of it, I think, is, you know, is there actually enough information in these data sets to be able to do that deconvolution well in the typical case? And I think actually there will be cases where you have really, really crowded regions where if there actually is variation in peak shape, which you have to assume basically there is, there's probably not enough information in one spectrum to do that. But if you train it across, you know, a large number, it kind of maybe can pick up some of these implicit priors, if you like. We were talking about this earlier on, you know, that maybe certain compounds in certain areas of the spectrum are more prone to wider peaks or different peak shapes, and in other areas it's more controlled, if you like. And so rather than having to explicitly encode that in some algorithm, even a simple one, you can do it more implicitly with this kind of neural network approach. At least that's my sort of take on it. But yes, these sort of, quote, simple deconvolution approaches, they work okay for some parts of it, but they're really not competitive in terms of most of it. Yeah.
Just on that point, and with GANs as well, I worry a little bit about the sort of hidden implicit biases that creep in. I mean,
Mm.
GANs are known to suffer from this feature called mode collapse,
Mm.
which is basically they only represent half the distribution.
Mm.
Because they're trying to resolve an ill-posed problem. So it's going to give you something which looks plausible every time, but there are the super resolution by its very nature has many answers.
Yeah.
You pick one.
Yeah, absolutely. And so, we certainly had, I mean, I mentioned very briefly sort of hallucinations and peak suppression. So one thing was small peak suppression, and we had, we weren't able to remove that. We tried really hard to sort of get around that. The other thing that we found, which I didn't really mention, was that we find some of the peaks seem to be sort of hyper resolved. So it'll resolve a broader peak into multiple peaks that we don't even see in our highest resolution experimental data. So for that, ultimately, we don't really know what the right answer is. We don't know if actually there could be more than one peak under there. Maybe if we had a brilliant, even higher field spectrometer, we could get to the bottom of it. Or the other option is, is it somehow a hallucination or giving you one result, but there are plenty of other results that would be consistent with the data. So, I think as with all this deep learning stuff, you have to be pretty skeptical about getting something for nothing. Okay. And so that, we were only able to validate it with the experimental data at different resolutions that we had. Yeah.
Okay, first of all, thank you so very much both for coming and all this fantastic presentation. Some questions I will reserve the right to ask you later.
Yeah, right.
Just some general things. So the last thing that you showed, the peak tracking, so you defined it as a problem, but I think it could be also a solution for it. It may be that your ideal acquisition condition contain quite a bit of overlap to begin with.
Mm-hmm.
And maybe this mobility can allow you this kind of, you're hidden now, you're being
exactly.
not hidden.
Exactly. Yeah.
So kind of.
No, you're absolutely right. I mean, I should have, that's thought had occurred to us as well. One thing is that I showed you a sort of pH gradient sequence, where the movement between these samples are a bit like a video frame. So, an object is moving from one side to the other. It doesn't move backwards and randomly backward and forwards. In reality, in the metabolomic situation, we don't necessarily have a pH gradient, but we can sort of introduce one artificially by taking a peak, measuring its position. And if it is only affected by one factor, like pH, which is an assumption in itself, we can then reorder the samples in that order. And we do get these nice trajectories. And you're absolutely right. You'll get some peaks that in one spectrum will be totally hidden and overlapped. And in other spectra, they appear. So you can then identify it, track it back into the overlap situation, and you'll get more information. So, that's just an example where I think, most of the methods that are out there for dealing with omics data just look at one sample at a time. And this is an example where we're looking at multiple samples and borrowing information between them to try and infer, basically solve some problems that wouldn't be solvable on one sample alone.
Yeah, no, there's pH driven, temperature driven, also don't necessarily affect all peaks in this.
No, exactly. No, some of them move really a lot, some of them don't move at all. pH is one, but the other thing that is a big factor in metabolomics is metal ion concentration, so calcium and magnesium.
The other question that I want to ask you is how essential is the symmetry for the J-res experiment? And I'm asking because that's what I want to talk to you about later on.
Okay.
Is that if you reverse the resolution problem, is that you have all the increments that you want, but what you don't have is resolution of the chemical shift, which is the case if you acquire data with a CKMG data. Is how well you can apply your method to gain the resolution on the chemical shift rather than on the J axis.
Yeah. Well, the short answer is the symmetry thing was a bit icing on the cake. Okay. So actually the network, vanilla flavor network retrained did pretty well on its own. And then adding that symmetry part to the loss function, no matter how much we weighted it, it never produced a huge amount of improvement, but it did produce an obvious improvement. So in the paper, we show some examples where, with and without the symmetric loss, you can see that there are changes in the symmetry of the data that is produced and things like that. But essentially, you as an NMR person, you might think it's adding a lot, but actually, I think most of the improvement is from the basic ESR GAN network, probably. Yeah. So, you know, we thought we were adding something nice. We were adding something, but it wasn't, you know, rocket science in that way.
All right, thank you.
Thank you so much.
Yeah.
I think we should,
Yeah.
but Ricky can come talk to us afterwards. Thank you everyone for coming. Thanks so much, Tim.
No worries. Thank you.